Question 10. Describe in basic detail how G proteins work. Include the roles played by GEFs and GAPs in this process. For some G proteins, GTP binding causes the protein to localize to the cell membrane. How does this work? All right, so we already covered this in a previous question, but I'll go into it again here and include explanation of what GEFs and GAPs do. All right, so G proteins basically have two states, a GTP-bound state and a GDP-bound state, and they cycle between these two states. All right, to get from the GTP state to the GDP state, the GTP gets hydrolyzed and converted into GDP and phosphate. All right, so G proteins are basically enzymes that have a GTPase function. So they can cleave GTP. All right, on the back side, going from GDP to GTP, the GDP does not get physically transformed into GTP. All right, this will require external energy input in order for that process to occur. Instead, G proteins undergo an exchange reaction. So the GDP leaves, and a new molecule of GTP comes in and binds. We'll see that soon. All right, there's the GDP leaving. And here's the new GTP coming in. All right, this is an exchange reaction here. All right, so now, this in a nutshell is how G proteins work. One other thing I like to say, the GTP bound state, that G protein is now turned on. Whatever its function is generally turned on in the GTP bound state. The GDP bound state is the off state, whereby the G protein gets reset. All right, now GEFs basically accelerate this exchange process. GEFs stand for guanine exchange factors. So they help accelerate the exchange process. GAPs stand for guanine activating or GTPase activating protein. So they activate the GTPase portion of this cycle. All right, now for the last part of the question here, how can GTP binding cause a protein to localize to the cell membrane? Well, there's some of these G proteins, or some proteins in general, that are covalently attached to lipid tails. It could be a fatty acid, as shown here, or it could be something like a farnesyl group. So one of the amino acids in the protein has a covalent bond to a fatty acid. All right. Well, there's some of these G proteins that are actually lipid-bound proteins. All right. And in a certain state, they can have the lipid basically pointing inward towards the hydrophobic core of the protein. All right, this is the GDP bound state. So when its function is turned off. All right, when they bind GTP, so when they undergo this exchange reaction, which is accelerated by a GEF, a guanine exchange factor. All right. Now they can bind GTP, all right, and undergo some sort of a conformational change. Well, in some of these cases, that conformational change can lead to that lipid now being pointed outward, outside of the protein, and it can embed itself into a membrane structure within the cell, causing the protein to now localize to the cell membrane.